Ashish and you're watching me on Medical India. So guys, today's lecture is very important and it is going to be the part of the AIIMS PhD entrance lecture series, and it is the kind of 11th lecture you can say of the series, and it is on the quantitative biochemistry, basically analytical biochemistry and all the kind of organic chemistry, physical chemistry, all the thing I'm going to give you an overview, and it is very important for the AIIMS PhD entrance. And apart from this is very important for also the ICMR JRF, DBT JRF and CSIR JRF gate also. Sometimes what happens in last kind of 6 months after giving the CSIR, ICMR JRF and DBT JRF, people have messaged a lot about to make the video on the kind of quantitative biochemistry. Why? Because it is the main part, and all the time 5 to 10 questions are asked in the ICMR JRF and this also the DBT JRF. So this is a very important lecture, and please guys watch till end, okay? And if you like the video, please share it to everyone from your juniors, from your kind of colleagues. You can send it, and please like the thumb button, please like the video if you are going to like the video indeed, okay? So guys, the main important thing here is the pH. So start with the pH. pH is equal to minus log H3 plus ion. H3 plus ion or H plus ion, that's the same thing. So the formula for the pH is given, and what happens in the kind of AIIMS PhD entrance and basically ICMR JRF entrance, ICMR JRF exam, they are going to give you the values based questions, direct questions. You are going to put the values and get the answer. So this type of questions are asked in the DBT JRF, ICMR JRF and AIIMS PhD entrance. Okay, so this is very important. Why? Because you can see that the product of the H plus ion concentration and the OH minus ion concentration becomes equal to the constant. So it is called the constant of water constant basically. So dissociation constant of water or constant water constant, okay? So it is equal to 1.0 into 10 to the power minus 14. So by this formula and the upper one, you are going to solve many questions, and the inverse also H3O plus ion, that is in the middle, concentration equal to 10 to the power minus pH, okay? So with these three formulas you are going to calculate the diverse kind of calculations for the pH basically in the exam. So guys, here I have listed some of the important kind of parts, or you can say cell organelles, cells and organs where the pH is asked sometimes, okay? So lysosomes has the pH of 4.5, mitochondria 7.5, stomach 1.0. Why? Because they are HCl. So intestine में ileum they it is 7.4, in intestine duodenum it is 6.0, and bile 8.3, blood 7.4, urine 6.0, and saliva 6.0 to 7.0, CSF very important 7.3, nucleus 7.3, HCl 3.1, not 1, okay? And washing soda will be 11.6 and the sodium hydroxide 14, okay? So these are the list, you can remember it. So guys, second thing is mole concept. Mole concept becomes very important. Sometimes what happens, they just ask the Avogadro's number value. What is the number in the Avogadro's value? So 6.02 into 10 to the power 23 is the actual number which is the present in the Avogadro number. So what is Avogadro number? Avogadro number is a kind of, you can say, just like you are referring the dozen, means half a dozen or this kind of thing. So this is basically the unit to measure the, or unit to dignify the what? The atoms, molecules or anything in the matter. So this is the quantity, or you can say the quantization of the matter in term of large number, because atoms are very small. So this is the number which is basically dignifies the flocks of some atoms. So this is 1 unit, so 1 mole. So mole concept is basically 1 mole equal to 6.02 into 10 to the power 23 atom, molecule or anything, okay? This is not just for the atoms. This is the big confusion among the people, okay? So this is not just for the atoms, it is also for the molecules, okay? So mole is equal to mass by molar mass. Very easy. So mass, so you can kind of thing you can use in the formulas. Sometimes they ask to calculate the mass, and sometimes while calculating the concentration you are going to get the kind of stuffs to calculate the moles. Why? Because moles are important in the concentration based questions. So basically this formula you can remember from here. So this is the kind of thing where they have shuffled the thing, moles equal to mass by molar mass, and everything they have shuffled, okay? And at the last you can see the units. For the molar mass it is gram per mole, and it is kg per mole in the larger amount, and the mass has the formula like units of g. So what happens, suppose if any atoms are there, okay, so what happens, the molar mass of that atom is equal to in gram. So in that gram, suppose sodium has 23 gram. So what happens, for sodium, what happens, there is the 23 gram of sodium has the 6.02 into 10 to the power 23 atoms. So in this way you can calculate these things. So you can say that in the kind of 18 gram, or you can say 18 gram of H2O, I am talking about ice, they have the 6.02 into 10 to the power 23 molecules of H2O. So in this way you can understand mole concept. Mole concept is very easy concept. Basically concentration based questions are very important. So normally here there are the 5-6 formulas, basically 5 formulas. These are very important. Molarity first is very important, equal to moles of solute divided by the volume of solution in liter. So very important, volume of solution in liter. Denominator is volume of solution in liter. But molarity is calculated in the solvent in kg. Okay. Here not the full solution is taken, but the solvent in kg. So moles of solute divided by the solvent in kg. Okay, so the mole fraction equal to x equal to basically moles of substance divided by total moles in the solution. So this is the mole fraction. So this is very easy, moles of the substance S divided by the total moles in the solution of that substance. Okay, so guys, normality equal to gram equivalent weight of solute divided by the volume of solution in liter. So this is the same thing, but what happened here, equivalent weight is taken. So equivalent weight equal to molar mass by n is the valence, or you can say number of hydrogen atoms in an acid or hydroxide ion atom in the base. So this is basically about is going to have the like idea concept about the valence and all. Okay, so this is basically the based on the valency. So guys, equilibrium. So what happens, any reaction is going on in your cell or anywhere has to achieve an equilibrium. Equilibrium means the rate of forward reaction equal to rate of reverse reaction. So what happens, see, this is the typical example where AA plus BB is going to give the CC plus DD. So in this case you can see that K1 and K minus 1. K1 is the forward rate constant and K minus 1 is the backward, or you can say reverse rate constant. So they have the values that is written here. K1 AA rate equal to K1, that is the constant, multiplied by the basically the substrates, and rate of reverse reaction will be the kind of rate constant of inverse reaction and multiplied by the product, multiplied concentration of the products. Okay, so at equilibrium the same thing I have said, that rate of forward reaction equal to rate of backward reaction. Okay, so you can calculate the like kind of equilibrium constant. This becomes very important. Here you can see in the right side that K equal to the product of the concentration of the products divided by the by the concentration of the substrates. So very important guys, you can remember this constant. This is very important, so very easy also to remember. So chemical kinetics and equilibrium. So what happened after the chemical kinetics have achieved, it becomes in equilibrium. So what happens, chemical kinetics of A plus B and is giving to C plus D, and here what happens, the rate of appearance equal to rate of disappearance of the product. So what happens, disappearance of the substrates and appearance of the products. Why? Products is going to be anabolized by the time. Okay, so there is an increase in the substrates and decrease in the product. So rate equal to minus the concentration of A or divided by T. With time the concentration of A is going to what, decrease, so the minus sign, and same with the product, they are going to increase, that is dC by dt. So this has the plus sign. Okay, so concentration symbol are in the super bracket. Okay, and T equal to time. If the reaction will be in that order, then the rate will be K C raised to the power the order, that is the N. Okay, so where K equal to rate constant, C equal to concentration, and N is the order. So you can here very easily see. So guys, here this is very important. the zero order, first order, second order, what is the basically half-life, and the unit which you are going to use. So please remember these things, these basically two things, the units and the half-life. So units of the zero order is mole per liter second, and for the first order, very important, many times these are asked, so it is per second, and for the second order it is liter, so that the inverse of the zero order. Okay, so half-life have the value of A naught divided by 2k, that is 2 into you can say the rate constant, and ln2 by 2, that is 0.693 by k. Okay, 1 by k, and the concentration of A naught. Okay, so initial concentration of A. So this becomes very important, half-life and units. So please remember these formulas. This is just the interactive lecture where I'm going to give you the information only. Okay, so please carry on that information in your notebook to the examination center, so that it is the last moment kind of revision. Okay, so guys, radioactive decay follows the kinetics of first order, very important thing. So what 0.693 by k formula is used. So no kind of any rush here, it is very easy. So guys, let's move toward the states of matter. States of matter basically governs the law, that is the ideal gas law, which is equal to PV equal to nRT, and all the symbols are written, and most of you all know. Okay, so basically here is important the rate constant. Rate constant has a value of 8.14 joule per Kelvin mole, or in the liter atmosphere it is 0.08206. So please remember these types of values. These are very important in ICMR JRF and DBT JRF. They actually ask what the Planck constant and all these things they ask, basically power on the electron, power on the neutron. In the ICMR they oftenly asks. Okay, so guys, please remember these kind of things. Boyle's law basically equal to PV equal to constant, that is P1 V1 equal to P2 V2. Okay, so it has the inverse relation. So if the pressure is going to increase, then the volume will decrease. So Charles' law, it states that pressure remaining constant, volume of a fixed amount of a gas is directly proportional to the absolute temperature. So here what the pressure is directly proportional to the absolute temperature. So Avogadro number is the relation between the kind of volumes of gas at the same condition of A and the number of molecules. Volume and number wala is given by the Avogadro number, and so is the ideal gas equation, that is PV equal to nRT comes into existence. After all these derivations, the ideal gas equation, the upper one, come into existence. So Dalton's law of partial pressures basically states that total pressure exerted by a mixture of non-reacting gases is equal to the sum of all the partial pressures exerted by them individually. So here P equal to P1, that is the pressure exerted by the molecule 1, then the molecule 2, then the molecule 3. So this is very easy. Just remember the relationships. Okay, the Charles law, Boyle's law, Avogadro law, and the Dalton's law. These three laws basically are important. Then comes the ideal gas equation. So guys, enzyme kinetics, very important for all the examinations. So guys, first graph you can see here that energy level is basically going to decrease by the catalysis with the enzyme. Okay, so what happens, there is a kind of decrease in the activation energy. Okay, so this activation energy is reduced and the catalysis occur by any enzyme. So you can see in the second figure that as the concentration of the substrates are going to increase, okay, then the rate of like you can say here is going to increase. Then what happens, the substrates are going to be depleted by time, so becomes the Km and the 1 by V max. Okay, so the maximum velocity, the half of the maximum velocity when plotted on the substrate concentration, it becomes the Km. Okay, and the equation which is going to be the part of it is the Michaelis-Menten equation, which is written here. So Michaelis-Menten equation basically becomes very important because some of the direct questions are asked from these topics. So please remember this formula. This formula is very important. So guys, basically here what happens, this is the Michaelis-Menten equation and this is the simple concentration rate graph. But when you extrapolate this graph, the inverse of basically the units, the parameters, then what happens, then comes the Lineweaver-Burke plot. So this becomes basically the extrapolation of the slope, and this becomes the minus k, okay, minus km, 1 by km. So this becomes very important. Here you can see 1 by km, okay. So this becomes very important. So this is double reciprocal, which is Lineweaver-Burke plot and the direct plots. So what happens basically here, to remember that competitive inhibition, and the best example here is the malonate wala and lactate dehydrogenase. So what happens here, the competitive inhibitions basically increases the km value and the Vmax value unchanged. In the non-competitive inhibition, Vmax decreases but the km unchanged. So please remember this. And in the uncompetitive inhibition, both Vmax and km are going to decrease. So this is very important. Many times asked in the like kind of CSIR, ICMR, JRF, DBT, JRF exam. So can be asked in the PhD entrance also. In biochemistry, this is the common question. So they basically what happens, this topic is going to come in the like kind of B part also, the discipline based. So please remember these things, okay. So guys, stereochemistry basically S and R system, SR system basically. So SR system may what happens, there is a kind of increase in the valency toward the anti-clock direction, and in the R system increase in the kind of clockwise direction in the valency, in the charge. So this kind of thing is called the SR system. So S and R molecules can be easily distinguished. So the second thing becomes the D and L type. In D type, the right-handed, you can see here, right-handed hydroxy group are there, okay. But in the L type, there is the left. So L means left, D means right. Dextrolevio you can say here. So dextrolevio, why? Because the chiral carbon becomes very important. By having the chiral carbon, it becomes optically active, any compound. So the C decides the chiral carbon. Chiral carbon means that carbon is bonded with all the kind of atoms which are different. So here you can see H is different, OH is different, CH2 OH is different, and COH is different. So C has all the carbon, all the atoms which are different from each other, okay. So this is called the chiral carbon. So the two important here concept is the apimer and anomer. Anomer, always remember the C1 position, okay. So anomer at the C1 position, and it is written as alpha and beta, okay. Alpha means right side OH, or you can say down OH, and the kind of beta means the upward OH. So you can remember in this way. And apimer is also very important here, where you can see that it becomes like kind of at the C5 atom, okay. So C5 atom gives the apimer, and apimers are basically the optically active, it is basically for the like kind of implementation of the optically active compounds. And basically these are the term, you can remember that D-glucose and D-galactose are the apimer of each other, you can see here, okay. So the apimers becomes very important. So let's move ahead, and this is the amino acids. So amino acids may three important questions can be asked, that is the rarest amino acid. You can see here leucine is rarest, then the serine, then the lysine, then the glutamate, okay. And the abundant are, sorry, it becomes like kind of reversed, okay. So please mind it, abundant is basically leucine, then serine, then lysine, then glutamate, and the rarest is tryptophan and greater than methionine and all. Two other amino acids besides the 20 traditional amino acids are solenocysteine and the pyrrolysine. So pardon for the like kind of mistake that has happened there, okay. So please remember that essential amino acid, all the essential amino acids are listed here: arginine, histidine, isoleucine, leucine, lysine, methionine, phenylalanine, and threonine, tryptophan, and valine. You can watch my video on the amino acid basically which is in the biomolecules part in the month of like October 2020. So there you can find all the tricks to remember the essential, non-essential amino acids and the conditionally essential amino acids, okay. That is semi-essential amino acid, you can say. So the amino acids can be classified hydrophilic and hydrophobic. Hydrophilic means the water loving, that is the polar, and it is negatively charged, positively charged, or uncharged. So again, hydrophobic can be aliphatic side chain with the aromatic side chain. For aromatic side chain, that is the phenylalanine and tryptophan, and for the aliphatic side chain, the hydrophobic one, it becomes glycine, alanine, valine, leucine, isoleucine, proline, and methionine. So negatively charged are aspartic acid and the glutamic acid. So aspartate and glutamate. So you can remember that both the acids are negatively charged, and all the three bases, like basic amino acids, are positively charged. Is the lysine, arginine, and the histidine. And uncharged, that is neutral, serine, threonine, asparagine, glutamine, cysteine, and tyrosine. Okay, so guys, these are the informations about the kind of amino acids. So guys, let's move toward the Ramachandran plot. You can just see that the psi and phi angles are basically plotted. Psi is the carbon bond, and phi is the carbon and nitrogen bond in the amino acid. So what happens, anti-parallel beta sheet, basically in the, they can ask like the orientation in the, like they will say that the which is, they can basically will give you to label basically the position of all the amino acids. You can see right-handed alpha helix at the lower quadrant, then in the upper quadrant parallel beta sheet and anti-parallel beta sheets, and you can see left-handed alpha helix and the like right upper quadrant. So please remember these three things: right-handed alpha helix, left-handed alpha helix, and the beta sheet. So these are very important. Collagen you can remember or remember not. So nucleic acids becomes very important. Here you have to remember the ribose and deoxyribose basically. So deoxyribose in the DNA and ribose in the RNA. Deoxyribose means the like deoxidation of the like ribose, where the what happens, the OH group may say O becomes, O is removed. Okay, so in this way the like dideoxy have the two O's, that is the two oxygen from the hydroxyl group will be removed. So guys, please remember these things, and you can remember the structure of the adenine, guanine, cytosine, and thymine. Sometimes it becomes very important, but you have to remember the pairing. Adenine will pair with the T and guanosine will pair with the C. So these kind of things you can remember. Okay, so it becomes very important. Purines are what? The adenine and guanine, and pyrimidines are thymine and cytosine. So how I have remembered basically purine and pyrimidine in my like kind of 12th, it by 9. See, ADE and 9, Gua and 9. So 9 means 9-membered ring, so the purine. So in this way you can remember. Okay, so purines are 9-membered ring, and 9 are adenine and guanine are the purines. So in this way you can remember. Okay, so guys, let's move ahead, and you can see here that the difference between the A, B, and Z DNA. So here what happens, A DNA basically you can see right-handed, B is right-handed, and Z is left-handed. And the second important thing here becomes the base pairs per turn. So in the A DNA 11, B DNA 10, Z DNA 12. So this becomes very important basically. And the third you can remember here is the kind of, you can say the axial rise. Okay, 0.26, 0.34 angstrom, 0.34, and the 0.45 nanometer. In angstrom that will be 3.4. Okay, and you can remember the twist angle, but this is not important. Okay, so they can ask about the base pair per turn and the orientation, that is the right and left-handed. So remember that left-handed are only Z DNA, that zigzag DNA. So carbohydrate metabolism becomes very important, and here is the chart which shows how many like kind of ATPs, NADPH, water, everything is produced in different steps of the carbohydrate metabolism. In glycolysis you can see that the 2 NADH and net 2 OTP is formed. Okay, so guys, in the transition reaction, that is the link reaction, 2 NADH is only produced, and in the Krebs cycle you can see that 6 NADH and 2 FADH, 2 ATP is produced, and as a total you can see that 4 ATP is produced in the like kind of as the ATP, and 10 NADH and 2 FADH 2, and the total 38 ATP is basically produced by all these processes. Okay, so you can remember it. And the second important thing here is between the difference between the anaerobic and the aerobic respiration. In anaerobic respiration you can see that the 2 ATP generated, and aerobic respiration 36 ATP net is basically generated, and gross if you will talk, then the 4 ATP and the 38 ATP. The final product will be alcohol and the or lactic acid depending on the like kind of tissue, and the lactic acid is produced in the muscle, as you all know, and the area where it occurs will be cytoplasm and the aerobic in the mitochondria. So please remember it. So guys, organic chemistry is very difficult to handle in the one class, so please remember the R groups. Basically in alcohols, what happens? OL, ethers, ether, aldehyde, L in the name, you can see the ethanol, diethyl ether, ethanol, propanone for ketones, and oic acid for carboxylic acid, and esters के लिए OH. Okay, so these kind of you can remember the suffixes. Okay, and the formula and the symbol has been shown here. You can analyze it, you can see it very carefully because it is very important for the examination. So vitamins becomes very important part of the biochemistry. For vitamins, there are two types of vitamins: water soluble and fat soluble. In fat soluble, they are A, D, K, E, and the common names of the all the like kind of vitamins are given here. Please remember it. Sometimes what happens, they ask the common name. So the water soluble vitamins becomes the non-B complex and the B complex. In the non-B complex, ascorbic acid, that is vitamin C, and the B complex is thiamine, vitamin B1. Please remember the common name, again I am saying. Okay, so riboflavin, B12, B2, niacin, B3, biotin, B7, pantothenic acid, B7. Okay, and in hematopoietic it is folic acid and the vitamin B12. Okay, so in pyridoxine, that is vitamin B6, pyridoxal, and the pyridoxamine. These are all the derivatives. Okay, so please remember the common names from this chart. Basically, not remember the kind of the classification. This is not very important, but the common names becomes very important. Okay, so let's move ahead and see the deficiency of the kind of hypoconsumption of these like kind of vitamins. Thiamine B1 causes the beriberi, riboflavin B2 glossitis, vitamin A basically remember what they causes, they causes night blindness. Okay, niacin pellagra, pyridoxine anemia, and B12 pernicious anemia, very important guys, very important. Folic acid B9 anemia, pantothenic acid burning feet, biotin nerves disorder, vitamin C scurvy, very important again. Calciferol, that is vitamin D rickets, tocopherol. These are the multiple kind of syndromes. Okay, fertility disorders basically in the females and males. Okay, so phylloquinone, vitamin K, basically clotting problems, so blood clotting problems in the vitamin K. So you can remember this kind of thing. This is sometimes be useful in the examinations. Okay, so this is basically the units to convert. Okay, so if they are saying the femto, they'll ask that what is the femto, then that is the 10 to the power minus 15. You can remember it, I am not going to dictate it. Okay, so guys, let's talk about drug dose relation. Drug dose relation become very important. Why? Because suppose if you are going to test the like dose of the drugs, so what happens, you have to give like different titers of or concentration of the drugs in the same animal. So what happens in the early stage, it becomes, suppose drug is not the kind of thing which is beneficial. Sorry, the like pardon for the family coronavirus, basically it is the default thing. Okay, so guys, so what happens, these basically becomes very important. Why? Because the drug testing is a kind of a rigorous thing where you have to like kind of shuffle with the many kind of titers or concentrations. So what happens, the lethal dose, if suppose drugs are not the thing which is basically like the delicious, you are going to eat it. Okay, you are going to consume it, so it is in a only threshold manner, it is very optimal. So this is therapeutic range, or you can say therapeutic range where it is effective in the like kind of the kind of thing, it becomes toxic effect and then the lethal effect. So ED50, that is the effective dose 50 at the 50% of the response in the like kind of individual, and TD is the toxic toxic dose at the 50% of the response, and LD is the lethal dose with 50% of the response in the individual. So these three terms becomes very important, and you can see in the kind of left side that therapeutic effect is seen first, and then the toxic effect, then the lethal effect. So ED50 and TD50, these becomes very important calculating the therapeutic index. Sometimes the numerical based questions are asked in the therapeutic index. So TD50 by LD, ED50 becomes the formula for calculating the therapeutic index. Okay, so please remember the formula. Sometimes what happens, the formula based questions are asked, which is just the putting the values in the formula and getting the answer. So please remember it. So this becomes very important, guys. So thank you guys, thank you so much. Please like, subscribe and share my channel, and I hope you have enjoyed the video. Thank you guys, thank you so much. And if you want to connect with me on the Instagram, Facebook and Twitter, then you can follow the kind of descriptions which is given in the text box. So guys, thank you so much.